G'day Junior Techies, and welcome to this video dealing with dihybrid crosses. Remember, this is just one video in a series of videos to better our understanding in genetics. So dihybrid crosses, we're looking at a genetic cross involving two traits. I've included an example here for us to work through just to get a better understanding of how a dihybrid cross would work. So looking at our example over here, we'll see that we have a plant species with two characteristics. We look at flower color as being the one trait, and then the plant height being the other trait. If we analyze our question, and a lot of the time it's all about our understanding of the context in which we're asked these questions. So if we have a look now, we'll see that the characteristics each have two variations. So with regards to flower color, the flower may be red or white, and the plant may be tall or short. This is going to be dependent on the allelic combinations at the end of the day. We can see that for plants that are heterozygous for flower color, they have red flowers, and this gives us a valuable context. This tells us that in the heterozygous condition, the dominant allele will be expressed, and in this case, for red flowers, it's going to be the color red in terms of flower color. In the homozygous recessive condition for plant height, the plants are short, which means the recessive allele is going to be for short plants. So understanding this context is quite important for us in determining the phenotypic expression of each of these in our dihybrid cross. So looking at the format in which we solve a dihybrid cross, it's very similar to the monohybrid cross, except now instead of having those four possibilities in terms of the combinations of the gametes, we have 16 possible combinations. But we start with the same format. Our P1, F1 generations, we look at writing in the phenotypic traits first, and we can see that our one phenotype is a tall plant with red flowers and a tall plant with red flowers on that side. If we look at the way that these genotypes are combined, we see we've got a heterozygous for tall and heterozygous for red flower, and on the other side, the other parent, exactly the same genotype. The reason I'm using this example in terms of the genotypes is that we get a very, very specific, unique ratio expression in terms of the phenotype at the end of this genetic cross, which I want to highlight for us. So if we break up our different gametes, or we break up our genotypes into the different gametes, we'll see that how we do this is we've got to combine for each of the parents the alleles in all the possible ways for them to be combined in the gametes. So meiosis results in the random combination of these alleles, completely random. So we'll see that for our first combination in the gamete, we'll have a dominant H and a dominant F, and if we continue to do it this way around for both of the parents, we're going to have the gametes form in the following way. Because we've got the genotypic combination the same for both parents, we'll find that the gamete combinations are going to be the same as well. We can proceed to put these into our genetic cross, and we do that here, and then we combine these alleles with one another as we complete our genetic cross. We're going to be putting them together in the following way, where we have the alleles for the specific trait next to each other in each of the blocks. So here we've done the first four combinations, and we can proceed to do the next 12. There we go. We've now completed our genetic cross, and what we're going to do is analyze what has come out of this. You'll see I've underlined the first combination there, where we've got a homozygous dominant and homozygous dominant combination of those alleles, which are obviously going to result in the dominant expression in the phenotype. But looking at how each of these have come together, I'm now underlining those where we have the unique combinations of the different alleles. And you'll see where you have a different color present each time is the unique expression or the unique combination. If we take this or these different combinations out and we place them into a genotypic ratio, you're going to see the ratio as it is written down at the bottom next to the genotype. Now it's up to us to look at each of these combinations and have a look at what the expression is going to be as the phenotype, keeping in mind that a homozygous dominant and heterozygous condition result in the same phenotype for the trait. So we're going to see here that we can count each of these. We've got to make sure they tally up to 16 to make sure we've counted out the possibilities that exist. And then we are going to, and my suggestion is use a highlighter to highlight all of those possibilities that produce the same phenotypic expression of a characteristic. I've done that over there, and you'll see we have 9 possibilities where we have the expression of the phenotype being red flowers, tall plants. Next, we have a look at the expression of red flowers, short plants, and you can see we've got 3 possibilities there. And again, we look at white flowers, tall plants, and finally, there is only one condition that will result in white flowers and their plants being short. If we put this out into the phenotypic expression, we'll see the ratio being 9 to 3 to 3 to 1. And this is a very, very unique ratio. When we have parents with heterozygous for each of the traits that we're looking to trace in the offspring, we're going to find this ratio every single time. 9 tall plants with red flowers to 3 tall plants with white flowers to 3 short plants with red flowers, and only 1 where we have the homozygous recessive for both of these being short plants with white flowers. So this is how we would solve the dihybrid cross. I hope this makes a lot of sense. Please keep in mind that this is one video in a series dealing with genetics, so please access the others to better your understanding in this way.